So now that we have got our VCF file, for example, in our folder, you can see I have made a file of folder of results, and in this result directory, I've got our VCF file, VFC file. So I'm gonna move to the results folder and then see if the file is present there. Yes. If we can just by any chance read our file, yes, it contains all the information now being represented there. So here we can see it contains all the type of information that we saw in our VCF file when we are looking at the presentation of our course. Now you can see that we have got our files, we have got our VCF file, and we now have our variants. So how to separate these variants, how we can separate the Indels from SNPs, how we can create these files. So there is a certain pipeline that you have to implement as well. So if you see that after the indexing, we can just extract the Indels. So what are Indels? You know that insertion and deletion sequences that are present. So we have to just design, conduct, and synthesize the Indels. So for the separation of handles, we just gonna use the VCF tools. View minus V is the feature for viewing the handles in our VCF file. So we are just going to do that. VCF, we have the VCF file and just gonna view the handles. Yes. So now you can just see that only the handles are being separated. Here you can see the handles. So for that, I'm just gonna make a new file, handles dot VCF file. So now that we can, if we can see if the file has been created. Yes, handles dot VCF file has been created. Cat handles.vcf. Now you can see all the file contain the handles data. Insertion and deletion mutations are present here. So one type of variants has been extracted from our VCF file mylab. Moving on, we have to just by any chance if we want to know that how many number of handles are present in your reference sequence, how many number of handles are presented there in your VCF file. So by using that same command, command again, you can just grab the number of. So there are 43,231 handles mutations that were present in our VCF files. So same for the our SNP file. Here we can see that if we got our single nucleotide polymorphism variations. So how we can separate these? By using again the SNPs view system, SNP view tool. We can just separate the SNPs from here. Here you can see we have just separated the SNPs from here. The SNP file is, now I'm gonna make a new file for it, snps.vcf. So yes, file is created. I got the snps.vcf file, handles.vcf file. Two type of mutations have been extracted successfully. So I think that was the main goal for our lecture, for calling the variants and then extracting out the variants. So now the remaining part is how to visualize these variants, how we can visualize these kind of variants. So copy number variations are different kind of variations that can be visualized in human genome as well. So if you want to do that, you just have to copy the same stuff and then go again like that. So for visualizing variants, we have to require these files. So for example, for if we are going to visualize the variants, for example, we are going to visualize the variants that we have extracted, we need two tools which are mainly used by now. So result of the variant caller can be viewed with the tools like Board, IGV or UCSC browser. Let me just open. I think we should cover this in the next lecture. So for this lecture, separating the SNPs and the Indels was enough.